Eagle Double G.
Snoop Dogg.
Da-da-da-da-da. You know I'm mobbing with the D.R.E.
Yeah. You know who's back up in this motherfucker. So blaze the weed up then. Blaze that shit up, nigga. Yeah.
Hi, I'm Sinead Noel McIntosh, and I'm here to tell you about mitosis. Mitosis is the process by which all eukaryotic cells divide. It serves to produce new identical tissue, whether for growth or wound repair. It's also how some non-eukaryotic cells asexually reproduce. Mitosis produces two identical daughter cells. Meiosis is the other form of cell division that eukaryotes use to produce sex cells, or gametes. These cells contain only half the DNA of the mother cell, where cells produced by mitosis contain all of it. While cells of any ploidy, haploid, diploid, or polyploid, can go through mitosis, meiosis only works with diploid cells to produce haploid cells. Another difference is that 4 daughter cells are produced, not 2. But we're talking about mitosis here. There are 3 main parts to the cell cycle: interphase, mitosis, and cytokinesis. The cell must pass through certain checkpoints to successfully and correctly divide. Let's discuss this further.
To really understand mitosis, we need to first discuss the cell cycle. When a new cell is produced, it must first go through G1 phase. G for gap. In G1, the cell is checked for size, nutrients, and a proper amount of organelles. G0 state can be viewed as an extended G1 state, where the cell is neither dividing or being prepared to be divided. If all is good in G1 phase, then the cell can go to S phase. In the S phase, S for synthesis, in this state, all the chromosomes have been replicated and are prepared for division. After the S phase, we can move to G2. G2 checkpoint checks for DNA, size, and if the spindle apparatus can be formed.
So to recap, in order for a cell to progress into the mitosis phase, it has to be big enough. It has to have produced enough organelles to be able to make 2 functional cells, have enough nutrients to sustain both of its daughter cells, have duplicated its DNA successfully, and it has to have formed a spindle apparatus.
Now the cell is ready for mitosis! When DNA is just chilling in the nucleus, uncoiled, it is known as chromatin. Little proteins, called histones, keep the protein coiled and small enough to fit inside the nucleus. During prophase of mitosis, DNA is coiled very tightly to form chromosomes. The combination of DNA and histones make up chromatin, only be seen under an electron microscope. The X-shaped thing that is often used to represent chromosomes is actually 2 separate but identical strands of super compressed DNA called chromatids. One of these was produced during the synthesis phase of interphase. Which one? We don't know, they're identical. These sister chromatids are attached together by proteins called cohesins, which are most concentrated in a region called the centromere. What's the centromere? It's
It's the region along the chromatids that spindles attach during mitosis. We'll get to that later. During prophase, chromatin coils into chromosomes, the nucleolus dissolves, and the spindle apparatus starts to form at the poles of the cell. We've already discussed chromosomes. The nucleolus is where ribosomes are produced in the nucleus. It's not included in the animation because people aren't perfect. The spindle apparatus is what will pull chromosomes to opposite sides of the cell by sending out microtubules. These attach to the centromere of each chromosome. Once the spindle apparatus is formed and the nucleolus is dissolved and the chromatin is coiled into chromosomes, the nuclear membrane dissolves, and this is what signals the cell to move on to metaphase. In metaphase, the chromosomes of a cell line up along the metaphase plate, exactly in the center of the cell. The spindle apparatus has attached microtubules to the centromere of each chromosome. They're attached by things called kinetochores. Kinetochores, which are complexes of proteins that hold microtubules to the centromere. Then the cell enters anaphase. Are pulled to either pole of the cell. In telophase, the nuclear envelope reforms around the chromosomes that have migrated to either side of the cell. Positioned around the center of the cell is a ring of actin proteins. During cytokinesis, this contracts and squeezes the cell, forming a cleavage furrow and then, finally, dividing them in two. Telophase is technically the final stage of mitosis. After this comes cytokinesis, when the cytoplasm in the cell is actually divided into two separate cells. Cytokinesis is the end of mitosis and the end of this video.
It's the motherfucking Eagle Double G.
Snoop Dogg.
You know I'm mobbing with the D.R.E.